This summer we're going to have a chance to cover only a portion of Chapter 17, and our coverage is going to focus in on a cycle called the Urea cycle. However, as you do scan through Chapter 17, and I advise you to just kind of flip through and look at some of it, you will notice that a big portion of Chapter 17 does cover concepts that we have already looked a little bit at. That is general protein turnover, how proteins can enter into catabolism, or how they can be built in anabolism. So it is something that we have already covered the highlights of. So without further ado, we want to jump into the specifics of the cycle called the Urea cycle. And perhaps when you do look at that cycle, you think about urine, right? And you're not wrong, because urea is the primary component of urine. Now, it is our body's way of getting rid of toxic ammonia. And depending upon the organism, in aquatic animals, they're able to diffuse ammonia, toxic ammonia, across their cellular membranes because immediately into the environment it is diluted. But we don't have that advantage or luxury, and so we have to convert a toxic ammonia into urea, which is a polar molecule that can then get transported within the blood to the kidney and can get excreted in the urine at that point. So this is our body's way of getting rid of toxic ammonia. And so maybe you're thinking, yeah, okay, so why would anyone want to drink their household ammonia cleaner? I mean, clearly toxic, right? Not something you want to have in your body. So the question becomes, why do we have toxic ammonia in our bodies anyway? And that's because it turns out that proteins themselves are toxic, right? Remember the amino group on a protein. So in excess, dietary protein has to be detoxified, and that it has to be deaminated, so that then whatever is left there in terms of the protein skeleton can enter into metabolism in a variety of ways, whether it be catabolism or some other modes that we'll talk about in a moment here. So these excess dietary proteins, they have to be deaminated, or as we talked about in extreme conditions where the body needs, say that in long duration exercise actually does end up using tissue proteins, those must be deaminated as well before they can enter into catabolism. So that amino group must be removed. That is what we call the deamination. And then that toxic ammonia, through a series of steps that we do call a urea cycle, is made into urea so that it can be eliminated from the body. So what that leaves us with then is, of course, the carbon skeleton of the protein. And we recognize that carbon skeleton, remember the reaction by which glutamate can get deaminated and become alpha-ketoglutarate. Well, that alpha-ketoglutarate, right, could enter into the TCA cycle. So we recognize a couple of ways in which those carbon skeletons can enter metabolism. We could also think about the way the glucose-alanine cycle and the very close conversion between alanine and pyruvate, right? That really a simple reaction there. So pyruvate can be synthesized from amino acid carbon skeletons. Of course, then that too can be used, depending upon the situation, to make acetyl-CoA, and if that is accumulating, we know sometimes that's used to synthesize acetoacetate, depending upon where the needs of the cell are. So a lot of ways in which these carbon skeletons can then be utilized, one of them, of course, being catabolism, stripped and ripped of its high-energy electrons, maybe within that turning TCA, made into fully worn out, thoroughly used up CO2, and of course water, if it is fully catabolized. But sometimes the body needs these carbon skeletons for other reasons, for perhaps anabolic reasons, maybe to synthesize glucose. And we know that pyruvate can certainly, and is, the precursor to the synthesis of glucose via gluconeogenesis. So amino acids we recognize too for their precursor metabolite ability in being able to make glucose in gluconeogenesis. Also recognize that if someone is starving or in those very long duration exercises, we'll see that those carbon skeletons can in fact be used to synthesize ketone bodies. And we talked about the way that can happen in certain extreme diets as well. So they can be used for synthesis in ketogenesis, or also in fatty acid synthesis, right? Two very different situations there that we would be thinking about. One would be accumulation of acetyl-CoA due to a slowed TCA, the inability to meet all the needs of the cell via just that catabolism of fats and the need for ketone bodies then. But versus fatty acid synthesis, well, that would be when the TCA was accumulating acetyl-CoA just due to excessive nutrients, and then perhaps this carbon skeletons, maybe in an excessive situation of taking in a lot of nutrients, and a lot of which were amino acids, then they could be used to synthesize fats. So this gives us an idea of the eclectic diversity of the fates of the carbon skeletons, and leaves us now in a place where we can follow up on what happened to that ammonia, right? That toxic ammonia that was removed first to make the carbon skeleton, and to follow the steps of the urea cycle. So we want to zoom in on the liver where the urea cycle is taking place and look at the nuances of the way in which urea is synthesized.
This is a big picture view of the urea cycle, and we're going to zoom in on little portions of it as we go. But for now, I want you to notice a couple of things. Notice first that part of the urea cycle takes place within the mitochondrial matrix. Another part of that cycle takes place within the cytosol. So we are actually looking at a switch in compartmentation throughout the cycle. We can also notice throughout the cycle that there are a lot of intermediates towards the synthesis of urea, and one of the things that one of our goals is going to be to watch where the nitrogens that get into that urea molecule come from, and also where the carbon atom comes from. Great. So we're going to see that as we go. We're also going to know as we go that this process, this pathway, is going to be endergonic. It's biosynthetic, it's anabolic, it's synthesizing something. Okay, let's zoom in on the first step of the pathway, beginning by looking at the fact that glutamate is oxidatively deaminated, so it loses its amino group, forming ammonia, and it's an oxidation as well, so it also forms reducing power, either in the form of NADPH or NADH. Let's zoom in on just this region of this cycle. Nice. So what we can see then here is that ammonia, toxic ammonia, is formed from that deamination event that takes place right here. And this ammonia is going to react with bicarbonate. And this is going to form the first intermediate of the urea cycle called carbamoyl phosphate. Carbamoyl phosphate is a very famous molecule because not only is it an intermediate towards the synthesis of urea, but it is also a starting molecule towards the synthesis of certain pyrimidines, so nucleotide biosynthesis, important there. It also, in organisms that don't have the urea cycle, it also is able to be used as a precursor to synthesize arginine, and you'll notice that arginine is the last intermediate towards the synthesis of urea, so if an organism doesn't make urea, it might use this molecule as a precursor to make arginine. Cool. So this molecule very important in numerous ways, and in fact representative of that importance is that this enzyme right here that catalyzes this reaction, we're not going to look in depth at it, but I want to mention it, carbamoyl phosphate synthetase 1. It is one of the most abundant enzymes in the mitochondrial matrix of the liver cell. It actually accounts for up to 20% of the protein content in the mitochondrial matrix, so it is a very, very, very prevalent enzyme. So we might write down then the ammonia released when glutamate is oxidatively deaminated reacts with bicarbonate to form carbamoyl phosphate. Step 1. Now look at something here. Notice that this is a highly energy consuming step, highly endergonic. It requires 2 ATPs to power the synthesis of carbamoyl phosphate. All right, let's look now at how carbamoyl phosphate is going to react with a somewhat unusual amino acid to form the next intermediate in the pathway. Here we have it, carbamoyl phosphate reacting with this unusual amino acid, ornithine. This is going to make another unusual amino acid called citrulline. So we see that being the next step of the process. Now notice that there is a transporter. In fact, this is a citrulline-ornithine exchanger and allows for citrulline to go out, ornithine to come in. It's interesting to note, actually, that ornithine, when it comes to the urea cycle, is really kind of a catalyst because it allows for the formation of the citrulline intermediate, and then at the end of the cycle, it itself is regenerated and comes back in and does the same thing again. So, if you will, it's regenerated but not consumed, just like a catalyst would be. So we're going to go on to the next step. One thing I want to note before we leave this too far behind is notice that one of the nitrogens that's going to be converted to and be found, if you will, in urea is originating from the ammonia molecule off of glutamate. So that's the origins of one of those nitrogens. Now, citrulline is going to swish out into the cytosol, and in the next step, we'll look at the fact that citrulline reacts with aspartate, most often that originated in the mitochondrial matrix and was exchanged for glutamate in the glutamate aspartate translocase. So aspartate and citrulline react with one another in another endergonic, very energy-consuming reaction that consumes an ATP. But wait a minute, it's more like consuming what? Notice it goes to AMP plus pyrophosphate, which this pyrophosphate likely goes to 2 inorganic phosphates. It's more like consuming 2 ATP to power the formation of this next monster of a sexy molecule called Argininosuccinate. Argininosuccinate is going to be converted in the next step to arginine. So we see how arginine becomes an intermediate in this pathway. Notice though that the next toxic nitrogen originates from aspartate. So one of them came from glutamate, ultimately from the ammonia there, and one of them came from aspartate. And we have now accounted for both of the nitrogens that will be found in urea that is excreted. Okay, so in the next step then, we see that fumarate is lost, arginine is formed, and in organisms that do the urea cycle, then this will be lost, this molecule or this moiety will be lost as urea. So we kind of come to the end of our cycle. So argininosuccinate is cleaved, forming arginine and fumarate. So the urea molecule, the carbon came from the bicarbonate in the very beginning. One of the nitrogens came from ammonia. The other nitrogen originated from aspartate, and we finally have a toxic urea that can now be excreted from the kidney. It's transported in the blood from the liver to the kidney and then excreted from the kidneys. And there you have it, the best way for us to get rid of toxic nitrogen. Now, before we leave this, let's make sure that we go ahead and keep track of all that's happened and write down all that's happened in terms of energy and whatnot. So the guanidinium group of arginine is cleaved, forming urea and ornithine. We already mentioned that. And now we'll go ahead and mention what our energy input was like. 4 ATP total, right? Notice here we had the 2 ATP input. Here we had an ATP, but it was hydrolyzed to AMP and PPI. So that's more like the equivalent of 2 phosphoanhydride bonds, 2 high energy bonds. So it's more like the equivalent of 4 ATP that are consumed here. 2 nitrogen atoms of the urea come from the ammonia right here and from the aspartate, and eventually here we have both of those. The carbon that is on the urea molecule came from the bicarbonate that reacts with the ammonia here. All right, that is the urea cycle in a nutshell, and now we have a better idea of what happens to that ammonia that is produced when protein is deaminated. Phew, I'm glad we're done with that. All that talking of urea cycle has made me need to use the little girl's room.